Welcome, everyone. I'm Dr. Carmen Pugliafito, and I'd like to welcome to Retina Synthesis Dr. Donald Fong. Don, how are you doing today?
I'm doing great, Carmen. Thank you for this opportunity to talk.
Don is Vice President of Ophthalmology Clinical Development at Annexon, which is a clinical stage biotech company located in California, and looking at a variety of diseases, including geographic atrophy. Don, could you tell us in a broad sense what the approach of Annexon is?
Yeah, sure. I'm happy to do that. Annexon's approach is focused on the classical pathway of the complement system. And the complement system has been shown to be involved with AMD. First, there have been genetic studies that have shown that the complement system is involved. There have been genetic subtypes where there is reduced complement. And when there's reduced complement, the risk of AMD decreases, and in stages where the genetic subtype shows increased complement, then there is increased AMD. So we know that the complement pathway is involved in geographic atrophy, and the complement system basically has three systems: the alternate pathway, which is essentially a spontaneous hydrolysis of CA. Of C3, and you get activation of the pathway that way. And then Annexon is focused on the classical pathway, which is a substrate-based activation of the complement pathway. And we believe that a substrate-based activation is a differentiated approach. And this is because in macular degeneration, you've got degeneration at the photoreceptors and the RPE with drusen and so forth. And what we see in animal studies and in human path studies is that there's activation of C1q in these areas. And our approach is to block the complement pathway by blocking the classical pathway at the very beginning at C1q.
So C1q is an important molecule in the complement pathway, and your animal studies showed that C1q blockade was related to photoreceptor rescue. Isn't that correct?
Yeah. So we have a bit of evidence on this. So if you look at C1q in mice and so forth, C1q is involved in neural development, in making sure that the neuronal synapses are adequately pruned. So it's very activated early on in neural development. And it's also activated with aging. So you build up C1q. And so it makes a lot of sense that if you want to block the complement pathway, you want to start with C1q. We also see C1q in light damage mice. So these are mice that have been exposed to bright lights to damage their photoreceptors, and you see C1q activation there as well. So C1q definitely involved in degeneration. And the other part about C1q is that macrophages that express C1q is located throughout the entire retina, and with this expression of C1q everywhere, it really does make sense to try to block it to prevent aberrant activation of the complement system.
Apellis is looking at blocking C3, and Iveric is looking at blocking C5, and they have ongoing phase 3 trials, and the Apellis results are forthcoming. How does your approach differentiate from blocking these two downstream complement factors?
Yeah, excellent question. You know, the complement pathway is very complex. There are many nodes where we could intervene. And the Apellis approach is to block C3 at the amplification pathway, and the C5 and the Zimura product blocks the complement pathway of the C5 lower down. So we believe that if you want to block the complement pathway, where you want to start is at the beginning at the classical pathway at C1q. And the reason you want to do this is because when you activate C1q, you activate the complement pathway. You're not just activating the complement pathway, you're actually preventing the buildup of opsonins. C4A is actually is a nephrotoxin. So if you have a lot of that around, that brings in a lot of phagocytic cells and so forth, and that could lead to continual inflammatory activity. C4B is an opsonin, which enhances phagocytosis. So if we block the complement pathway further on, you're not blocking all the production of opsonins and aflatoxins that are being created through activation of the classical pathway from C1q activation. So we feel that that blocking the classical pathway with C1q will more fully inhibit the classical pathway and prevent the other products from building up. So that's one of the advantages we believe, is that it's a fuller blockade of the complement system. And some of the other advantages with our product potentially is that because it's blocking the classical pathway, it's not touching the amplification pathway, allowing normal cleavage of C3 and C5. And what this does is that it allows normal homeostasis of the complement pathway, which is necessary in retinal health. There was an article from NIH that showed that C3 actually is necessary to prevent the continued inflammation from degenerating photoreceptors. I think we all know that with time and with age, we get photoreceptors that get worn out and they get digested, and there's waste products. And you really need to have C3 around to get those products removed. So, you don't necessarily want to fully block C3 to allow the normal homeostasis of the retina to proceed. Well, and one final advantage of the product that we're developing, ANX007, is that there is potentially an opportunity to do every other month dosing.
So going back to C3, if you're at the top of the complement cascade, isn't your molecule blocking C3 as well? So
C3 is part of the amplification pathway. So you have the classical pathway activating C3, you have the leptin pathway, which essentially is like the classical, but reacting to the mannose sugars on cell membranes, and you have the classical all feeding into C3 activation. So if we block the classical, the leptin pathway continues to work, the alternate pathway continues to work, and it's this allowance of the normal activity of those other pathways, which we think could add to some safety benefits to just blocking the classical pathway.
Mm-hmm. So your molecule is an antibody fragment, and what's the evidence that it blocks C1q? Do you have any evidence from your phase 1 studies?
Yeah, excellent question. We do actually. We've done phase 1 studies looking at injection of our ANX007 and then doing aqueous samples to look at the amount of free C1q. And about 17 such patients in our phase 1 study. And at 29 days, there's full blockage of C1q. There's no free C1q at 29 days after injection. So very good blockage.
Why do you think it might work longer than 1 month?
It's a good question. Our animal models show that when we model the data, that it's around for that much longer. And that is a question that our phase 2 study, which is ongoing, will look at, whether the every other month dosing will be as effective as the every other month.
So can you tell us about the phase 2 trial? It's a big trial, more than 200 participants. Can you tell us about the study design?
Sure. So it is a global trial. So we have sites in the U.S. We have 45 sites in the U.S. We have 10 sites in Oceania, which is Australia and New Zealand, and we have patients randomized 5 milligrams every month, and also to 5 milligrams every other month, and compared to sham. So a patient coming into our study essentially has 2/3 chance of getting an active agent in our trial. We do a lot of testing at baseline, and we see the patients every month. And our primary endpoint is at 12 months, and we plan to follow these patients for additional 6 months after. The study enrolled its first patient earlier this year, and we're on track to completing enrollment in the first quarter of 2022.
Mm-hmm. So how are you measuring geographic atrophy in the study?
Yeah, good question. The FDA actually has provided us some background, what they're willing to accept. And so we're looking at the autofluorescence as an endpoint. So we're hoping to see a reduction in progression of area that's lost, as identified by autofluorescence.
Are you doing other tests like microperimetry or OCT angiography in any of the eyes?
Not on everybody. Microperimetry is an interesting question. That's a way where you can correlate visual function with what you see in the retina. The problem with microperimetry is that it's time consuming, and some patients find it really uncomfortable. And when you have patient performance issues, that creates a lot of noise. Mm-hmm. And so we decided not to include it in our phase 2 study, but certainly something we can consider as we move toward the phase 3 studies and other studies.
Mm-hmm. So in the Apellis trial, a number of eyes developed choroidal neovascularization. So how does your study address the issue of choroidal macular neovascularization?
Yeah, excellent question. It is true that certainly in the Philly study, there was an increase in choroidal neovascularization. And I'm assuming that the same thing is going to be seen in the phase 3 study, given how similar the study design is. One thing that's different, as we've been talking, is that our approach is the classical pathway, and we're blocking it high up. And so when you block it high up, it does two things. One is that you may need less drug. It's kind of like stopping the train at the station rather than trying to stop it as it's running away on the tracks. So potentially our study uses less drug. It may be easier to inhibit it, the complement pathway. And the other issue which we talked about before is that because we're only blocking the classical pathway and allowing the other pathways to continue, there is potentially better retina homeostasis and better retina health. And this might lead to less choroidal neovascularization, but this is something we'll need to see as the results come in.
Are the eyes that develop choroidal neovascularization in your study excluded, or do you treat the eyes and move forward?
Yeah, good question. Well, we believe that the wet AMD or neovascularization is pretty well treated right now with a lot of agents. We feel we can manage this. And if patients end up developing this, we would expect that the patient to continue in our study to get our agent and treatment for CNV with an anti-VEGF agent.
Yeah, that's the approach that Apellis is taking in its phase 3 trial, which is different than what they did in their phase 2 trial, where they excluded those eyes.
That's right. But we now know that, wet AMD is very well treated. And, we have a treatment for wet AMD, but we don't have one for dry AMD. And so, I, as an investigator, it seems you want to be able to offer the patients a treatment for which none exists, and just control the side effects where it's already been shown to be well treated with existing agents.
Well, this is a very exciting and I think promising approach. Before we finish up, I'd like to ask you a couple of personal questions. You had a long career as a retinal clinician and as a very active clinical researcher, including some path-breaking work on the epidemiology of diabetic retinopathy. What attracted you to a career in industry?
Yeah, excellent question. I think that I've spent a lot of time. I've been in academics. I've been with a large group practice. I've done a lot of work with diabetic retinopathy. I started clinical research at my old company, Kaiser Permanente, and I started our ophthalmology program. And I think that as I age and think about doing other things, I wanted to have a different kind of impact. I've always had a public health approach to taking care of patients, and this is just sort of the next step in public health and ophthalmology in my career.
Well, certainly there is an epidemic of geographic atrophy that is accelerating because the aging of the rapid aging of the population, particularly over the age of 80. So this is indeed a public health challenge.
Yes, it is, and I think that we know a lot about wet AMD because there's a treatment for it, and with dry AMD, there isn't a treatment, and so there are potentially, many times the number of patients who haven't been identified, who haven't been referred in for treatment. Once we get a successful treatment, we're going to see probably more patients coming in to our practices with dry AMD.
Do you think that there will be a treatment for intermediate AMD anytime within the next decade?
Yeah, I think that's an excellent question, and I actually think that that's where the treatment needs to be. I think that geographic atrophy is basically death of the retina, of photoreceptors and RPE, and you really don't want to be intervening at the time of death. You want to try to prevent death, and so I think that the impact is going to be preventing patients from getting to death. And when things are dying, it's very hard to stop. It's much better to try to prevent the photoreceptor death from coming on, and that's kind of how the thinking behind the company. And I also want to mention, and I've forgotten this, is that the one other advantage with the Annexon product is the neurodegenerative protection that we get with our product. C1q, our C1q inhibitor, can prevent the deposition of C1q onto the photoreceptor synapses. So this can prevent complement-mediated neurodegeneration that is often seen in diseases like Huntington's and ALS and potentially Alzheimer's. So this is one other advantage of blocking at C1q. So you get the protection, but allowing the normal homeostasis, allowing C3 to proceed normally, and you get the potential neural protection by blocking of the C1q at the synapses.
Well, that's great. Thank you so much for joining us today on retina synthesis, Dr. Fong, and we look forward to some exciting developments from Annexon.
Thank you, Carmen. It really was a pleasure to talk to you.